Hey there everyone, welcome back to another video. In today's video, we are going to be looking at Biology Paper 6 Food Test. Now, this is also a way you can easily get free marks if you look at it, because it's something that's repeated in almost every single Paper 6. Okay, so once you're able to know all of them efficiently, you can answer them and get the marks. Now, the most repeated one and the most common one they ask is the test for reducing sugar. Reducing sugar is basically glucose. And now the reason they ask it so many times is because many people just write Benedict solution and then leave the answer like that. That will just get you the 1 mark. In order to get the second mark, you have to say heat it as well. So the test for reducing sugar is Benedict solution and then you heat it. Okay, now the positive test is basically the color change to over here it says red, orange, yellow, or green, but you can always tip to brick red. Okay, so for reducing sugar, blue to brick red. Now, another one that's commonly asked is for starch. Now, in order to test for starch, you go with the iodine solution. Okay, iodine solution tends from brown to blue black when for a positive color change. Okay, then we have vitamin C. For vitamin C, we use DCPIP. DCPIP. Now, you do not need to know the full form of it, so don't stress yourself learning that. The result for it is that it decolorizes. Now, over here, though A over here stands for accept. Okay, it says accept colorless, clear, or transparent. Please, let's just stick to decolorizes so that we are 100% sure that we are getting the mark. Then we sometimes also get protein. Protein, to test for it, you have to use burette solution. And the positive result for that is blue to purple or lilac. And you can see that this was added in a question. State the result for a positive test for protein, which is lilac or purple. And what else do we have? We have ethanol emulsion test. Okay, ethanol emulsion test is done for fats, lipids, oils, which are basically the same thing. Now, what you do is that you add ethanol and then you add water and then you shake it. Now, someone will just write add ethanol and test for this. No, you have to say these three things. Over here, though it's only for 2 marks, this can come for 3 marks, where 1 mark is for add ethanol, then add water, and then shake it. It's very important you know these three things because it can be really tricky if you don't know them. And oh, one more thing. Now this is for carbon dioxide, and this isn't asked that many times. Like you'll see very few questions on them, to be honest, but you should know them. So you can either use a hydrogen carbonate indicator which turns yellow, or you can or to be honest, just stick with lime water, which becomes cloudy or milky. Okay, stick with lime water. Now, let me just tell you for hydrogen carbonate, this one. This is an indicator used for carbon dioxide. When it is at normal carbon dioxide concentration, it's red in color. When it is at high carbon dioxide concentration, it's yellow in color. Okay, and when it is at low carbon dioxide concentration, it's purple in color. That's something extra that you should know. And oops, I think that will be all for this video. Let me just show you this one as well. Now, you might think that, okay, this comes from maybe just 1 or 2 marks. But look over here. Over here, they ask for the test for the fruit juice of starch. That's 1 mark here. Then reducing sugar, another mark here. Then the test of a positive, another mark here. Then fruit juice contains vitamin C, effect of temperature on vitamin C. So that means you'll have to somehow include DCPIP in this as well. So that's 6 marks over here. And that in total is 9 marks, which so 9 out of 40. 9 marks out of 40 can determine whether you get the A star or not. So that'll be all for this video. Make sure you're subscribed because I have one more video on graphs and everything coming really soon. Thank you for watching, and the link for these notes are in the description, and I'll see you in the next video. Till then, ta-da.